Well, Suzanne Rowe is now our next speaker, and she's going to be talking about methane genomics from the Pastoral Greenhouse Gas Research Consortium.
Thanks, Neville. So I'm standing in front of a body of work that's been completed probably over the last 10 years by John McEwen. I don't know what he's done recently, but he's suddenly become camera shy, I dread to think. There's only 2 names on the front of the slide. That's just because John and I put the slides together. There are heaps of people involved, and I'll try and acknowledge them as I go. So start by just describing the problem, or the opportunity, as we should call it. So our government has committed, I should say your government, our government has committed to reducing greenhouse gases by 50% of 1990 levels by 2050. So this is the minus 50 by 50. This is going to be very difficult. It probably won't be achieved, which means that there'll be a carbon price and methane will become expensive. This is very important in pastoral systems and in countries like New Zealand because about 30% of the methane actually comes from ruminants. So we can't go in and say industrial processes have to clean up their act. We have to do something with ruminant emissions, and biological systems are a lot harder to clean up. The good news is that methane has been shown to be heritable and repeatable over time, so it looks as though we can breed for low methane, and as geneticists, that's very good news. So genetic change can also be measured and tracked, so it's auditable. That's a very good thing for government, because they obviously need to audit it. Genetic change is often very low cost, it's permanent and cumulative, so it's sustainable. And we've got existing infrastructure. We already breed animals, we already select ruminants, so if we can infiltrate the system and put methane within it, then we've got a method to move forward and lower methane emissions. And this is basically a pipeline of work. So firstly, we need to know that there's heritability, that there's genetic variation that we can select on. We need to know if we're selecting safely, that if there are links to production traits that they're either neutral or favourable. We need to know if we can detect and measure methane, so we need to know if there are genetic and phenotypic markers for low methane that we can identify and then select for. And we need to implement this. We need to get this out to industry. So there needs to be some sort of breeding scheme design, and it needs to be implemented in existing breeding systems. So this is just a description of the work, and I'm probably going to deliver the punchline within this, and then I'm going to go through a whole heap of experiments and just a whistle-stop tour of some of the results. So the program started by taking central progeny test flocks and screening them for high and low methane emitters. And this was done in a respiration chamber. So animals were adapted to a lucerne pellet diet. They were put into crates and eventually put into respiration chambers for 48 hours. And methane emissions are measured constantly on a constant diet. After the 48 hours, there's a 14-day period of rest, and the animals are measured again. And that's really important, because we really want to know if we measure methane on an animal, and then we put the animal out to pasture or on a lucerne pellet diet, and we come back and measure that animal again, are we going to see the similar results? Is that trait repeatable? So that's what was done. These flocks were screened, and from this point, the top and the bottom, it was 100 size, I think, and 1,000 progeny. And this resulted in selection lines. So the high methane animals were selected upon using breeding values that were methane divided by dry matter intake. So we know that a big animal will emit more methane. What we really need to know is that residual yield of methane. So this is the high line, this is the low line, and after a generation or so, we're looking at a 9% difference in methane emissions. So we know that the difference between selecting for low and selecting for high on roughly 100 ewes, we achieve 9% difference, so we know it's heritable, we know we can select for it. What we need to know now is what we've done. So methane accounts for about 8% of the waste of energy from the rumen. And if you perturb a system such as that, you really need to know what the biological implications are and what you've been doing. So the rest of this talk basically goes through the difference between these lines. They've been measured annually in the respiration chamber and saying, what have we done, what's going on biologically, and there's some really interesting hypotheses we can draw from that. So this is just to say that the trait is heritable and repeatable. We look at methane in 2 different ways. We look at total emissions, but as I've said, what we're really interested in is methane yield, is the amount of methane that an animal emits given the amount of dry matter that it ingests. And we have moderate heritabilities. Methane yield is on the low side, but the heritabilities are significant. And what's really important is that if you measure an animal over a couple of days, then the repeatability of the record is roughly 0.9. So we know that if we put an animal in the chamber for 2 days, that the second day you're going to get a roughly same measure as you did on the first day. And if you measure them a fortnight later, the repeatability drops to around about 0.6. So really important, what's happening to other production traits. If we select for methane, what are we doing? And here we can see that pretty much there's not much going on, but there's a really interesting correlation with fleece weight. And we think what's happening is that once you perturb the rumen system, you're possibly changing volatile fatty acids and the way the animal utilizes them, and that there's something going on here with the utilization of those acids and wool production. So this is actually good, because this is saying that you get more wool for less methane. So we went on and looked a bit further and said, okay, so if we've got breeding values for these animals, what happens if we look at the breeding values for the low line compared to the breeding line values with the high line? And what this says is that low emitting animals, in actual fact, have more than $3 more breeding worth than high emitting animals. They're positive for growth, they're positive for adult weight, for reproduction, disease, wool, slightly negative for DAGs. But what we do see, and what we have a watching brief on, is survival. So it looks as though the low emitting animals are efficient, they're leaner, they grow faster, but it looks like there may be a slightly negative trend for survival. It's not significant, but we're watching it. So moving on, we're perturbing the rumen, so it makes sense to measure it. And this is work carried out by Wendy Bain using CT scanning and just looking at the volume of the rumen, whether there are physiological changes, and the difference in draft liquid and gas. And these are the results. And basically they're highly significant between the two lines. And what we see is that a low emitting animal has a smaller rumen, it has a faster throughput, and it looks as though there may be some other morphological changes, so they have more papillae in the rumen, and so it looks as though somehow they're compensating for that faster throughput with a different type of absorption. So then we thought, well, we need to look at what's coming out of the rumen. So we looked at volatile fatty acids, and again, highly significant differences between the lines. And volatile fatty acids were measured both in rumen fluid and in blood plasma. And what this shows is that, one, volatile fatty acids are very heritable, and two, that there's a big change. So low emitters tend to produce more acetate, and there's a different propionate to acetate ratio depending on whether we breed for high or low methane. So this is interesting because once we know that we can measure methane in a methane chamber, measuring methane in a chamber is really expensive. So we really want other predictors. We want things that we can go out, to the animal and make tests that we can predict methane and perhaps use for selection. So this is one of the sort of initial pieces of work. We thought, well, hey, we've got a predictor here that might be a lot easier to measure than sticking an animal in a box for 2 days. So from here we moved on to the actual rumen microbial composition, and this is where we look at the microbes within the gut itself and use a marker gene to sequence and group them. And what we're saying is, okay, we know that it's a bacterial fermentation in the rumen. We know that archaea in the rumen are what are producing this methane. So if we actually look at that composition, maybe it's controlled by the host, and it should be different for high and low emitters. And indeed, that's what we find. We see a very clear pattern between high and low emitters. We see that if we take these compositions and we shrink them down in some way, and all we've done here is take microbes and look at the way they plot, realize that it's quite planar. We can reduce them into a couple of dimensions purely by looking at the relationships between them. And when we reduce, we have this, this CA1 and CA2 are a representative of the way that animals plot over here. And we see that actually these dimensions are heritable. There are distinct patterns between high and low emitters. And that this coordinate here, this CA2, in actual fact, is highly genetically correlated to methane, and in particular to methane yield. So another exciting example of how we might be able to predict methane without having to go through really expensive experimental profiles. So in conclusion, if we look at highs versus lows, we know that after a generation that we had about 8% differences. We know that low-emitting animals had smaller rumens, faster throughput, a very different propionate to acetate ratio. The genetic correlation with short-chain fatty acids was around 0.8. The genetic correlation with different microbial structure was, it looks somewhere between 0.6 and 0.9, but I have to put a caveat there. We've only measured a small number of animals, so I'd hate for you to go away thinking we'd solved the issue. So then we moved on to think, well, if we take this measure over time, we know that there's diurnal variation in the way a ruminant eats and in the gas that it emits. So we really need to know if we're measuring at the right time. So we took each 5-minute measure from the 48-hour period and correlated it back to a 24-hour measure. And we actually found that the correlation is very high. So what that tells us is if we go in and take a quick 1-hour measure, we're capturing most of the variation. So we don't need to strap an animal in the box for 2 days. And that's a really important result. So ingenuity abounds. This is a portable accumulation chamber for a sheep. This is a chamber that's been traveling around the country measuring methane in sheep. And basically it's a perspex box with a water seal that we can stick a sheep in for an hour, put an eagle on the top and measure the gas emissions of sheep. And that means that we can go around and we can measure commercial animals and we can measure animals much faster, but most importantly we can measure animals off pasture. So we can start looking at the systems that we want to select within. And we can repeat in different seasons, ages and feeding values, feeding levels. So that was, again, another important development. And excitingly, when we look at the heritabilities and the repeatabilities and the genetic correlations that we saw from respiration chambers, they're very similar in these portable accumulation chambers. So it looks as though we have a decent proxy that we can use. This is another validation on pasture using SF6, and I'm not going to dwell on this, just to show that basically differences between the lines were very similar when we validated with this technology. And this is just another way of saying, okay, does it work on pasture? It's okay to find these differences on pellets, but that's not the New Zealand system. Go. So Ken's described the method for genomic prediction, and we use the same method. All these animals were very densely genotyped, so we used a mixture of high density and 50K, and they were all imputed to high density, and it was natural that we should look at genomic prediction. It's an expensive trait, and what we found was that when we took animals, trained in the parents and predicted in the progeny, then we get an accuracy of around 0.5 for absolute emissions and 0.46 for methane yield. So it looks as though there's certainly promise there, and given that a lot of the key sires within New Zealand have genomic prediction values, then plugging this into the index is feasible. So in summary, methane emissions have been found to be heritable and repeatable. Low emitters look to be similar or potentially more productive. They do have smaller rumens and they have a different rumen microbial community. They have lower volatile fatty acids or lower acetate in the rumen and plasma, higher propionate. But it looks as though we can go on and measure this. We can possibly use these predictors. We can use repeated brief measures, which are more flexible and cheaper than using a chamber. We've validated on pasture. There's potential for genomic prediction. And what we're moving on to is industry implementation in sheep. And to look and see if we can transfer and validate these learnings to dairy cattle and deer. Which, if these predictors work in sheep, and obviously sheep are much easier to handle and measure, then possibly we can transfer some learnings here, because we don't really want to put a deer into a respiration chamber, because that's going to be, That's going to be a cross-technician. So I'm just going to end on this, and this just basically shows the way it all fits together. We had these high and low selection lines, measured them in respiration chambers, looked at feed quality, looked at repeatability, measured carcass and alimentary tract, just to make sure, you know, see what we were doing and make sure that we weren't doing anything that was detrimental to the animal. We could then go and test other predictors on pasture and in the commercial population. We can link all these records to each other, and we start to get a link into commercial phenotyping. Once we have genotypes, we can have breeding values, and that moves us on to the all-singing, all-dancing sheep. So it's just really nice the way it started with the central progeny test animals. The same phenotypes have been gathered, so it all links right through. And because methane is something that's, you know, really important within the rumen system, we're actually coming up with other traits that are just as important to production as methane itself. And what that means is if there is no carbon cost, if carbon was zero tomorrow, methane still gives us a handle on what's going on in the rumen, on the volatile fatty acids, and very importantly, methane and CO2 emissions actually give us a really good indicator of feed intake. And, you know, and we've heard about feed efficiency and feed intake, so it's been a really interesting trait to be involved in. And, oh, somewhere there should be a picture. I've lost my picture. Oh, there they are. Of all the main protagonists. And the camera-shy John McEwen.
Thanks, Suzanne. Do we have any questions just for points of clarification? Thanks very much, Suzanne.